So a very good evening everyone. I'm Sahil Srivastava, currently studying at UCMS Bhairava, and today the topic is going to be one of my favorite from immunology, and that is actually complement system. So yeah, let's get into it, and we will discuss a lot about it, all the clinical cases and all the different mediators and inhibitors. Okay, so yeah, first we will write the definition, what are actually complement system and what are complement proteins. Okay, so complement system is, so complement, pardon me for my writing if you can understand. Okay, this system refers to a system of soluble and cell bound proteins that occur in normal serum and are activated by antigen and antibody reaction. If you aren't interested in all this definition, you can actually jump up to the pathways. Okay, so the term was actually coined by Paul Ehrlich. Okay, Paul Ehrlich coined the term complement. And I will give you another definition of it. Okay, so another definition would be a system of hepatically. I want you to write it down if you have copy or something, because it might help you in the longer run in actually all those USMLE or PLAB if you want, or NEET PG2 or your own country's exam, because these notes are actually derived from the step 1 book, first aid. If you know that book, it's actually a very beautiful book. Okay, so this definition: system of hepatically synthesized plasma protein that play a role in innate immunity or inflammation. So maximum of most of the complement proteins are synthesized in the liver, except the C1, which is actually synthesized in the intestinal mucosa. Okay, and there are going to be some properties of I will write some properties of complement protein. So first of all, they are actually very heat labile protein. Okay, heat labile protein, they are very heat sensitive. If you put them at 56 degrees Celsius for 30 minutes, they will get destroyed. So these are this one fact more: 5% of mammalian proteins are actually complement proteins. Okay, these are some of the definition. It might be overwhelming for you to remember, because yeah, okay, actually complement proteins. So without any further ado, we will actually jump into the pathways, because they are more important for us. So see, there are actually 3 pathways of complement activation. I will be covering all the important things associated with complement proteins, complement activation, sorry. Okay, so the first would be everyone knows the classical pathway. Okay, the classical pathway. See, I will tell you how to memorize it at the end of this video. Okay, and the alternative pathway, or sometimes it is also called as properdine pathway. You can actually jump at pathways if you are finding it boring. Lectin pathway, or you can say the MBL mannose binding lectin pathway. So see, first we will now we will discuss it individually. First is the classical pathway. Okay, so what happens in the classical pathway? See, let me first make you clear about immunoglobulins, because if you don't understand immunoglobulins, it will be hard time you memorizing all where the complement system binds and all. Okay, so these are 2 heavy chains of immunoglobulin, and this is 2 light chain of immunoglobulin. So this area is this area let me draw it again. Okay, 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 a second. Okay, this is FAB portion and this is FC portion of an immunoglobulin. For instance, let's take it as IgG antibody. There are actually 5 GAMED if you want to remember. Okay, IgG, IgA, IgM, IgE and IgD. Okay, so IgG, in IgG this is the FAB portion and this is the FC portion. In this FAB, the antigen comes and binds to it. For example, let's say there's an endotoxin, it comes and binds here. Okay, and it gets activated and then it will form an antigen antibody complex, and this will lead to activation of complement. The first complement, which is C1 in classical pathway, will come and attach at the FC portion. You can remember FC for complement, C for complement, but the C actually in FC portion defines the fraction of crystallization, if you want to know. But you can remember it as where the complement gets attached. It attaches with the FC portion of the immunoglobulin. So let me draw a beautiful diagram here. I'm going to describe like how the complement get activated. So let's take an example. This is a bacteria, okay, and let's see this. See, always remember bacteria are antigenically mosaic, means there are a lot of antigens present on bacteria. Okay, so let's imagine an antibody binds to it. Okay, let me make it better. You can understand. Oops. Make it better. Okay, take another color. Some good color probably would be it's too hard. Okay, so yeah, let's take an example that this is an antibody. It gets attached to the antigen at FAB. So the first complement protein will come and bind here. This is C1. Okay, C1 has 3 parts: Q, R and S. C1s is also known as C1 esterase. Okay, I will tell you what is the function of this. Okay, first let me tell you, C1 gets attached to the complement by the C1q unit. It is the recognition unit of C1. So this is Q. Okay, it gets attached to the C1. It gets attached to the antibody through C1q. Okay, and then the C1s will actually break up the C4 protein into 2 parts, C4a and C4b. So let me make a very basic diagram. So let's imagine this is a boy, okay, and there is a football up at his head. So let's imagine this is the B portion and this is the A portion. Whenever it gets activated by an enzyme, okay, then the A portion goes away and so let's see, it's sleeping, okay, and the B portion, B for boy and A for, you can say, apple, okay, and the boy gets activated. Yay. And now he will form the complement cascade. So see, C1 comes and the C4, okay, the C1, okay, and another, we actually denote the enzyme as C1 dash. Now this is an enzyme which will break C4 into C4a and C4b. We say this C1s, okay, and now another complement will come and attach to it, which is C4b. Okay, this will cleave the C4 into C4a and C4b, and now C4b will cleave the C2 into C2a and C2b. I will tell you what these things are going to do and in later part of this video. And then C2b, okay, let me tell you something more. C4b and C2b are actually called C3 convertase. These protein are going to convert C3 into 2 parts. They will cleave C3 into 2 parts, C3a and C3b. So here comes the C3 protein, and it has been cleaved into C3a and C3b. Now another protein will come, okay, and C4b, C2b and C3b together, they are called C5 convertase, because they will cleave the C5 into C5A and C5B. C5B and C5A. So let me tell you something very controversial here. This step C2A and C2B. Okay, this step differs in a lot of book. Some say the C2A comes and join the complement cascade, and C2B goes and acts as an aflatoxin. But what I will tell that C2B is actually going to come to the coagulation cascade. It differs from book to book. If you follow the Ananta Narayana book, you will see here is C2A. Okay, here is C2A and here is C2B. But if you follow the first aid book, okay, assembly step 1, you will see C2B. There's historically it was said to be like C2A is attached, and C2B goes, but now they have changed it. The protein that actually attaches to the complement cascade is C2B. Okay, so if you want to check, you can just go and check it online or somewhere else. But yeah, and C2B, C2A will go. And now see, we had these protein divided into two parts. Okay, C2A and C2B, but now all the proteins that are going to come will have only one unit. And like now we can just go numerically. C5 and then C6 comes, C7, C8 and C9. And let me tell you something, these proteins from C5 to C9, from C5 to C9 are also known as membrane attack complex. What they are known as? Membrane attack complex. So let me show you a diagram. So let's imagine it is C5B, this is C6, C7, C8, this is C9. They will form a cylinder type of structure. Sorry. Okay, let's make it again. They form a cylinder type of structure. Okay, let's see, C5B, C6, C7, C8 and C9. And this is known as membrane attack complex. So what they will do, let's imagine this is a bacteria. Okay, and this membrane attack complex will just fall down to the cell wall of bacteria. So let me tell you this, it is a gram-negative bacteria. Mainly membrane attack complex affect the gram-negative bacteria, and in gram-negative bacteria also the Neisseria species, which is meningococci and pneumococci. So see, this will like make a pore inside the cell wall of bacteria, and all bacteria's electrolyte will come outside, and the bacteria will eventually die. Okay, this just the membrane attack complex, what it does, it's just there will be like this loss of osmotic stability of bacteria. So you can imagine if a person is suffering from deficiency of any one of these proteins, the membrane attack complex will not form. Okay, if the membrane attack complex will not form, then they will be frequently suffering from this infection, mainly the Neisseria species, in which meningococci and pneumococci are integral parts. So let me tell you something more about this. The classical pathway. Okay, the classical pathway is actually antibody dependent, mainly to IgM. So how will you remember that these two antibodies were taking part? So here's a mnemonic: General Motor Mix classical cars. IgM activate the classical pathway. So let's actually recapitulate what we just studied. Let me take a good color. Okay, so what happened, C1 comes first, and from the Q unit, Q, which is also known as recognition unit, it attaches, and then C1Q, let me tell you something more, C1Q actually activates C1R, which activates C1S, which is also known as C1 esterase, and this C1 esterase cleaves the C4 into two parts, C4A and C4B, and then this C4 gets attached to the C1, and now the C4, which is also known as C4 dash, is known as C2 convertase, because it will convert C2 into two parts, C2B and C2A. C2A and C24A will go somewhere else. I will come to that later on this video, as I told you earlier. So now the C4 and C2 will activate the C3, cleave the C3 to C3A and C3B, and C3B will, like these three will cleave, C4B, C2B and C3B will cleave C5 into C5A and C5B. That's why C4 is also called C2 convertase, and C4 and C2 are C4B and C2B are called C3 convertase, and C4B, C2B and C3B are called C5 convertase. And now the numerical order follows. C6 will come, C7 will attach, C8, C9, and these five from C5B to C9 are also known as membrane attack complex. So I hope it was easy for you, and now we will actually move forward to the second part. Let me tell you something more. The first part here is there is requirement of calcium ion Ca++, okay, and here Mg++. So if you want to know, just know it there. I don't think this is as important as this pathway is, and don't forget these five: membrane attack complex 5B, 6, 7, 8 and complement protein 9. Now we will move to the second pathway, which is the alternate pathway, or we called it properdine pathway. So we will be moving okay, and let me tell you something first about this pathway. It is actually antibody independent. Okay, so here let me tell you, antibody doesn't directly activate the complement system. We can say antigen and antibody complex will, okay, leave it, I'll tell you some part afterwards. And let me first tell you what are the substances that activate it. Let me take another color. This color is being monotonous to me. Yeah, green, my favorite color. So many substances actually activate this pathway, and I will just list some of them. You can say bacterial endotoxin. Also, endotoxin are something mainly released by gram-negative. Some gram-positive bacteria also release it. So yeah, and there are cobra venom and all, but you don't have to remember it. You have to just go through your textbook and see it. I'll tell you here. It will be very time consuming. So yeah, let me start. Let's again make a diagram. This is another this is a bacteria. Okay, this is a bacteria, and so the first protein, let me tell you something. C3b is actually moving in our bloodstream, but it doesn't activate any complement because it is inhibited by two factors. First is factor I and factor H. Okay, these two factors don't let C3b to form the complement cascade. If it somehow forms the complement cascade, it will make the membrane attack complex and damage your own cells like RBC or something. But these two are there to inactivate the C3b. But when endotoxin or something comes in contact with C3b, it gets activated and binds to the endotoxin C3b. C3b will first come and bind with the complement. With the endotoxin, and then another protein, which is B, protein B. B will come, and the B part of it binds, and A part of it goes. Now, it will again. These both will form C3 convertase, and they will again bind one more C3B, and C3A parts goes away, and then another C3P. Then comes the C5B, and it is actually same after this. See, all those pathways come and unite at the same place somewhere, Okay, and C6, C7, C8 and C9. Okay, and these 5 will make membrane attack complex and kill this bacteria. So let me again repeat it. C3b attaches, then there comes B, A part goes away, B becomes the fat protein B, and then C3b again comes and attaches to the complement cascade. C3A will go, and then C5b attaches, and then 6, 7, 8 and 9, and see this all will form the complement cascade. So that's all from alternative pathway. Now we will move forward to the last pathway, which is mannose binding lectin pathway. As you know, mannose is a carbohydrate. Mannose binding lectin pathway. So what happens is, imagine this is a bacteria and this is mannose residue on it. This is a mannose residue. See, there is a protein known as MBL protein, mannose bind lectin protein, which is an acute phase reactant. When you are suffering from any inflammation or something, it comes and it comes into action. Okay, and let's say this is a bacteria who is doing an inflammation, and this MBL protein will come and bind to it. Okay, and what happens? There are two subcomponents, if you want to remember: MASP1, MASP2, which stabilizes and helps MBL to attach on the mannose residue. So what is MASP? Let me tell you. It is MBL associated serine proteases. So what happens? That this MASP1 and MASP2 and MBL, they form a complex and they somehow resemble the C1 of the classical pathway. See, now from here, okay, from here onwards, the other complements that are going to come are same as the classical pathway. If you have remembered the classical pathway, it will be easier for you to just remember the mannose binding pathway. So let me tell you. So there will be what was there in the classical pathway. First C1 and then C4. Sorry, C4. Okay, C4b and C4a will go. Then comes the C3. I'm sorry. Then comes the C2. C2b and C2a will go, and then there will be C4. Sorry. Aha. Okay, let me again repeat it. Then first C1, then C4, C2, and then C3. C3a will go, and then comes the C5b, C5a will go, and C6, C7, 8, and 9. And this 5 will form the membrane attack complex again, and it will cause the osmotic imbalance or the osmotic shock in the bacteria, and the bacteria will eventually die. So this was actually all. This is the pathway. It is also antibody dependent, if you want to remember. There is no use of antibody here. Okay, antibody doesn't directly activate the complement system. And now let me tell you something. I had told you earlier I had earlier told you that C2, the A part of every complement protein, in C2A, C3A, and C4A, these all act as anaphylatoxin, and these anaphylatoxin help mediating inflammation. And some of them, and let's take the classical pathway. For example, the C1 comes, then the C4b, then C2b, and C3b. Let's forget about the A part for now. This whole complex, okay, particularly the C3b, helps in opsonization. What is opsonization? It is the process, okay, it is a process in which actually facilitates phagocytosis. So for example, see, this is a neutrophil, okay, and it has CR receptor here, these teeth of it, and this bacteria is not so good now because it is going to be eaten. So C3b comes and attaches here, and it will cause the phagocytosis. So see, what opsonins do is, from one side they bind to the antigen, and from another side they bind to neutrophils or other phagocytes, and they facilitate the phagocytosis process. Now see, if someone has like grabbed you from one hand and grabbed someone else from one hand, they can like just slowly close their arm and they can like just make you to come nazdik what is in here. Okay, so yeah, that's what opsonin proteins or opsonin molecules do. Let me give you another example. IgG also is an opsonin molecule, and C3b. Don't forget these two. These are opsonins. They help in opsonization. Okay, and coming back to what the A proteins did. Let me take another color. It's being monotonous again. Okay, blue would be good. Yeah, so for your okay, it's raining heavily. Okay, this is the nucleus. This is the mast cell. Okay, and these are granules of mast cell. Let me enlarge and granule. So what happens? There are receptor C3a and C5a receptors on mast cells. And what these receptors do, the C5a protein comes and binds to it, and it causes the degranulation of the granules of mast cell. Mast cell actually like they are modified basophils, and they release a lot of chemical mediators or chemokines or cytokines, but the main here is going to be histamines. And as we all know, histamines are a potent vasodilator, and they increase the vascular permeability. So they what these C3a and C5a do, they just helps in degranulation of histamines from mast cells, which mediates the inflammation. And other important works of like a part of complement protein was, see, this is a HIV virus, and these are its spikes, GP for instance, GP 120. So what these do, they come and attach on a cell, which is a normal healthy cell of a human being. Here is a CD4 receptor. They will come and bind to the CD4, and they can easily go inside the normal cell. What is the work of the A parts? They come and attach to these spikes, to these GP120 spikes, and they help in neutralizing the virus. These are actually these are important things related to complement protein. So let me revise you all again. Okay, this is a diagram from Ananta Narayana. So let me revise you again. See, the classical pathway, there will be antigen antibody complex. C1q will bind and it will activate C1r, and C1r will activate C1s, which is also known as C1 esterase. C1 esterase will then activate C4. It will cleave C4 into C4a and C4b, and C4 it will attach with C4b, and then it will cleave C2 into C2a, C2b. I have already told you that C2a and C2b is a controversial topic. In like earlier time, it used to be like C4a will join the complement cascade, but nowadays if you will see the first aid book or any other updated international edition book, you will find C4 to be here, but I will let it be. Okay, let it be. I won't just do anything. And this is called as C3 convertase, and C3 convertase will cleave C3 into C3a and C3b. Okay, and C3a will go and act as anaphylatoxin and cause the degranulation of mast cells. Okay, and then C3b will activate the C5, like this C4b, 2a, 3b. Okay, they will activate the C5 and cleave C5 into C5a and C5b, and then they will form the membrane attack complex, which is C5b will attach to C6, C7, C8 and C9, and this will form the membrane attack complex. And in the lectin MBL pathway, MBL will attach to the mannose on pathogen surface with the help of MASP and MASP2, mannose associated serine proteases, and then C4 will be converted to C4A and C4B. C2 will be cleaved into C2A and C2B, and C4B2A will act as C3 convertase, and they will finally do the same. The C3 will then convert into C3. C3 will be cleaved into C3B, C3A, and C3B will convert the C5 into C5A, C5B, and C5. Then the numerical sequencing C5, 6, 7, 8, 9. They will form the membrane attack complex. And about the alternative pathway, it actually happens due to the spontaneous hydrolysis of C3 and deposition of C3B on pathogen surface. I told you C3B will be moving into your blood, but they are inactivated by two proteins. I told you two factors, two proteins, protein H and protein I, or factor H and factor H and factor I. They will inactivate this C3B so that it doesn't cause autolysis of your own cells, particularly RBCs or some endothelial cells. And then factor, if there will be pathogen, it comes and binds to the pathogen, and factor B will come and attach to it, C3B, and the B will be cleaved into two parts, BA and BB, and this C3B will form C3 convertase, and C3 convertase will again cleave the C3 into C3A and C3B, and they will again form this C3BB3B, and then C3BB and 3B will form the membrane attack complex. Now the C5 will come, and 6, 7, 8, 9. So this is the gist of the complement system. So let me tell you more about let me tell you the clinical correlates. So there is an enzyme, okay, there is a protein. It's actually something clinical related to it. There is a protein known as C1 esterase inhibitor. So what it does, it will inhibit the C1 esterase. Okay, so it inhibits the C1 esterase. So what I told you during the classical pathway, that C1 esterase, okay, C1 esterase was cleaving C4 into C4B and C4A. So what it does, C1 esterase inhibitor, I will denote it from C1SI. So let's see, this was C1SI inhibitor. So it was inhibiting the C1S protein, so that C4B and C4 was not cleaved into C4A and C4B. And now, as you can see, if we have inherited deficiency of C1, if we have inherited deficiency of C1S inhibitor, then if it isn't there, let's say this isn't here, so no one is there to stop C1 esterase from converting C4 into C4B and C4A. So this will again lead to the formation of membrane attack complex, which it will lead to the complement cascade activation, which will lead to the formation of membrane attack complex. And this membrane attack complex will come and lyse your own RBCs, or say other endothelial cell. Okay, so if we have C1S inhibitor deficiency, then we do suffer from a particular syndrome known as angioneurotic edema. I have brought some photos for you, so you can see there's edema here, and one of the causes of this angioneurotic edema is the deficiency of C1S inhibitor. And I would like to tell you, just go through the book once, Vishwanath Narayana, or you can use the Apurva Sastry, or if you don't want, you can go to the first. That would be all from my side for today. Thank you and have a good day.